Yeah, the Emperor's funny, it's like, we should, I guess,
All right, hello everyone. Welcome to another session of our Evolution Seminar Series. Today we have a very special Early Career Awardee Seminar. This award is made possible by contributions from the College of Agricultural and Life Sciences, the College of Letters and Sciences, the Nelson Institute for Environmental Studies, and the Graduate School of UW-Madison. Our very special awardee today is Maria Kalambokidis from the University of Minnesota, working in Michael Travisano's group, bringing an ecological and evolutionary framework to our understanding of origin of life studies. So without further ado, let's get to the fun part. Please join me in welcoming Maria Kalambokidis.
Thank you. Hi, everyone. It's so great to be back in Madison. I actually did my undergrad here several years ago. And so I've started with a photo of the Galapagos Islands that was taken during a study abroad trip as an undergrad. And so it's very full circle, great to be back at the place where I became really excited about evolution. And in particular, I've been interested in big questions in biology. So Earth so far is the only planet that we know of to harbor life. And not only does it harbor life at all, but it harbors an incredible diversity and complexity of organisms that have changed dramatically over time. And a big goal of evolutionary biology has been to explore the processes of diversification and complexification. And we found that evolutionary outcomes result from the interplay of adaptation, chance, and history. Adaptation being the result of natural selection, chance due to random processes like mutation and drift, which can have long-term consequences known as historical contingencies. And because of this interplay of random and non-random processes, evolution is often unpredictable or unrepeatable. But nevertheless, if we look back at the history of life on Earth, we can see a general trend of increasing complexity, beginning with the emergence of prokaryotic life, the origin of eukaryotic life, from which evolved complex multicellular life, including all plants and animals, and even eventually complex social interactions like eusocial insect societies or cultural transmission. And what you can see when you look back at the history of life on Earth is not that evolution proceeded in a linear fashion. It didn't always lead to greater complexity, nor did it lead to the exclusion of simpler prokaryotic life. But what you can see is that the life that evolved later in Earth's history could not have emerged at the outset. And so prior evolution enabled the emergence of greater complexity. And one framework for understanding how complexity can evolve is known as the major evolutionary transitions, which is the idea that there were pivotal moments in Earth's history where a new level of individuality emerged that then transformed the adaptive landscape on Earth. And among these transitions include the origin of life, the origin of eukaryotic life, and multicellularity, among others. And there are many common features amongst these different major transitions, including that they all involve individual replicating units that come together to form a new reproductive unit. So for example, single cells becoming a multicellular individual. And this transition is possible given that there is sufficient cooperation within this new unit such that it can overcome competition between groups. And so this interplay of cooperation and competition within and among groups means that eco-evolutionary processes are key to understanding transitions in complexity. This then leads us to the question of how can we study these major transitions, given that some of them occurred only once in Earth's history, so the likelihood of recreating them is very low. Many of them occurred billions of years in the past, and so it's difficult to get full information about them. And so our approach is to use experimental evolution to explore the relevant processes that might contribute to or hinder these transitions in complexity. And my thesis in particular uses 3 different systems to study 3 different transitions in complexity. The first I'll be talking about is the origin of multicellularity. I'll then touch on the origin of eukaryotes, and then use those to describe our approach to the origins of life. So beginning with the origin of multicellularity. So for those of you who aren't familiar, in the Travisano lab, we have a system where multicellularity emerges very quickly in response to a selection regime known as settling selection. And the idea is that when we propagate our cultures, rather than propagating a well-mixed culture into fresh media, we allow an aliquot of that culture to sit on the bench for 7 minutes, allowing some of the cells to settle before transferring just the bottom portion of the tube into fresh media. And so we're selecting for the ability to settle faster through liquid media. And so since multicellular individuals are bigger and can settle faster than single cells, one of the results of this work is that multicellularity emerges in response to settling selection. This selection regime has been repeated in a variety of systems, including 2 different species of yeast, Cerevisiae and K. lactis, both of which evolve multicellularity in the same way. And because we're in Minnesota, we call them snowflake yeast. And so far, all of these experiments have been done in monocultures, so just a single species. But I wanted to add just one layer of ecological complexity and evolve them in co-cultures, the 2 species together, to see how interspecific interactions can shape the transition to multicellularity. And fortunately, we can distinguish between unicellular and multicellular individuals based on colony shape and between the 2 species based on colony size. So the first thing I found is that when you propagate these species together without settling selection, the species coexist and remain unicellular throughout the experiment. So shown here, as unicellular Cerevisiae is shown in teal, unicellular K. lactis is shown in black. And you can see that throughout this 19-day experiment, they begin and remain unicellular and continue to coexist. And this coexistence actually was not initially obvious that this would happen, given that we thought perhaps they occupy very similar niches. And so to understand why they're able to coexist, I did a spent media assay where I grew each species on their own and each other's spent media, and found that K. lactis is able to grow better on the Cerevisiae spent media, and Cerevisiae is able to grow better on the K. lactis spent media. And so this indicates that the species can either utilize a byproduct of the other species, or they're preferentially using the resources in the environment in different ways, such that they occupy sufficiently different niches, allowing for coexistence. So that's what happens when you don't have settling selection. With settling selection, we found that K. lactis becomes multicellular first, which allows it to rise in frequency and sweep through the populations. So multicellular K. lactis shown in pink. And this happens whether you're beginning with a 50% initial frequency of K. lactis or 25 and 75%. In all of these cases, K. lactis emerges first and rises in frequency. And so now you can see that species coexistence depends on the tempo of the transition to multicellularity. So the evolvability of K. lactis, the ability for it to become multicellular first, provides a competitive advantage and allows it to increase in frequency. Now it means that species coexistence depends on this tempo because in some of the conditions, for example, 3 out of the 6 populations, when beginning with a 50% initial frequency, Cerevisiae appears to be excluded by the end of the experiment. Same with the 25% initial frequency, and in 4 out of the 6 populations with a 75% initial frequency. And so you can clearly see the effects of competition with K. lactis on evolutionary outcomes for Cerevisiae. But you also see the effects of competition with Cerevisiae on the emergence of multicellularity in K. lactis. So here we have the frequency of multicellular K. lactis in the monoculture shown in light purple and in the co-cultures in the other colors. And you can see that when Cerevisiae is not present, so in the monocultures, multicellularity emerges sooner and rises in frequency faster compared to the co-cultures. And this is so competition with Cerevisiae slows the rate of the emergence of multicellularity for K. lactis. And this is likely due to the greater competitive ability of Cerevisiae when the 2 are grown together. So you can see that when over 24 hours of growth, S. cerevisiae is able to invade from rare without settling, whereas neither species has a greater competitive ability when settling. But the transition to multicellularity then shifts these dynamics. So now multicellular K. lactis can invade from rare and has a greater settling ability compared to multicellular S. cerevisiae. So not only did interspecific competition shape the transition to multicellularity, but the transition to multicellularity itself then shaped the nature of the species interactions. And this is an example of an eco-evolutionary feedback where ecological interactions shape evolutionary outcomes, which then shapes the nature of the interactions. So looking back at our major transitions framework, we can see that adding just one layer of ecological complexity, so 2 species instead of 1, dramatically alters evolutionary outcomes, which underscores the importance of ecological context in shaping the transition to multicellularity. So at this point, you might be wondering, okay, K. lactis can become multicellular first, which of course would give it a competitive advantage before S. cerevisiae ever even becomes multicellular. So what happens if the two are both multicellular at the same time? So in a different experiment, we did invasion from rare experiment for when both species begin as multicellular. And you can see that multicellular S. cerevisiae is able to invade from rare in both conditions, and the invasion dynamics of K. lactis are frequency dependent. However, in both cases, when they both begin as multicellular, regardless of which is rare, they can coexist throughout the experiment, showing that the tempo of the transition, if it were similar such that they were multicellular at the same time, would allow for coexistence, which isn't always the case if they're both unicellular. But if you look closer at these dynamics, it shows you an indication of what we also saw in the competition assays, which is in the initial 10 days of the experiment. So before adaptation potentially altered the dynamics, we see that S. cerevisiae mainly only in rare invades in the control conditions, and the opposite is true for K. lactis, invades in the settling conditions. And this is likely due to the greater settling ability of K. lactis due to its ability to flock together. So I said that both of the species become multicellular in the same way, and that is through a staying together mechanism of when the cells are budding, they don't bud apart, and the daughter cells remain attached to the mother cells, which creates these chains in this snowflake phenotype. But there also is a coming together mechanism at play where K. lactis, unlike S. cerevisiae, the multicellular clusters are able to stick together, which forms even larger clusters and allows them to settle even faster. So not only is potentially within group competition and cooperation and between group competition important for these transitions, but cooperation across biological scales. So between cooperation might also be a layer of the ecological context that could influence the transition. So we've looked at what happens if they both begin as unicellular and if they both begin as multicellular. Wouldn't it be interesting to see if they can become multicellular at the same time? So concurrent emergence of multicellularity, how does that shape evolutionary outcomes? And you may think that this is not possible because the species have the tempo that they have. But it turns out that the snowflake phenotype arises from recessive single locus mutations that undergo loss of heterozygosity. So S. cerevisiae, which is diploid, requires homozygous recessive genotype in order to become multicellular. The heterozygotes and the other homozygotes are both unicellular, which delays the emergence of multicellularity compared to haploid K. lactis, which only requires 1 mutation. But what previous work of Dr. Bea Baselga-Savera, who is a postdoc in the lab and an incoming faculty member, she found that the heterozygotes, while unicellular, become multicellular faster because they're already halfway there. So we can harness this to then it likely has a similar tempo of the transition to multicellularity compared to K. lactis. So when I ran the previous experiments of the co-cultures, here's a 50% initial frequency of K. lactis with homozygous S. cerevisiae. I did the same thing with heterozygous S. cerevisiae, which can become multicellular faster. And you can see it just like seriously screws up the dynamics. It's completely different. you can see that multicellularity does emerge for S. cerevisiae. It's shown down here in dark purple, a little difficult to see. And it emerges at about the same time, but it really alters dynamics. It's unclear what's happening. And so our future directions are, I'm continuing this experiment for much longer to see if the frequencies stabilize over time. So that is the work so far for studying the origin of multicellularity. We're now going to shift directions and discuss a different system that might potentially be relevant for eukaryotes. So, oh, I just realized I'm on a different slide. Sorry. So for those of you who aren't familiar, the last eukaryote, or you probably are familiar, the last eukaryotic common ancestor was formed following an endosymbiosis of a bacterium in an archaean. And we're interested in how this initial association potentially could have formed. And all of the work that I'm about to describe is, of course, done in collaboration with my advisor, Mike Travisano, but also with a postdoc in the lab, Dr. Naui Medina Chavez, who is our local archaea expert. And she's done a bunch of work, both growing and working with archaea. And so together, we've collaborated on how to establish a system of archaea and bacteria that can be grown together and therefore describe how the initial association could be formed between the species or between the taxa. But at this point, I want you to forget about the origin of eukaryotes momentarily, forget about major transitions, because unlike the multicellularity work where we already had a pre-existing system where we had observed the transition to multicellularity, that does not exist for eukaryogenesis. And so we had to establish a new system in the lab. And in doing so, we sort of developed a different project that allowed us to understand the adaptation more generally of these species before then growing them together and discussing eukaryotes in a little bit. And one thing we learned is that a big limiting factor for growing archaea in general is they are either difficult to culture or grow very slowly. And some of the fastest species that grow are halophilic. So we ended up embarking on an experiment looking at the adaptation of halophiles more generally for both archaea and bacteria. So central to the history of the study of archaea is their discovery in a variety of extreme environments. An interest in these extreme environments has only increased recently, given their potential relevance for environments beyond Earth. And this has led to more and more studies exploring the limits of life. So how does life adapt to survive in these extreme conditions? And what are the limits beyond which life cannot exist? And key to understanding that question, of course, is that adaptation is context dependent. So the ability to survive in these extreme conditions depends on a whole host of factors that are both inherent in the environment itself, as well as the history of the organism. So if we look at just one environment in particular, for example, a salt lake, we know that there is at least one environmental component that is considered extreme. There might be others. In this case, high salinity, but this is just one environmental axis. There are others, such as the amount of other resources in the environment and just all other abiotic and biotic factors that would influence an organism's ability to grow. And importantly, adaptation along one axis, for example, to high salinity, is influenced and constrained by the other environmental components. And therefore, adaptation within one particular set of conditions might lead to the emergence of fitness trade-offs in a different set of conditions. So to how this question has typically been explored, of what are the limits of adaptation in these environments, has been to sample from a variety of extreme and polyextreme environments and characterize the microbial communities found there. And this work has been extremely informative for understanding these communities, inferring functionality, but it can be challenging to observe evolution as it unfolds, given that they're a description of a community at a particular time. And it also can be challenging to disentangle the roles that the different environmental components play during their adaptation. So our approach, once again, is to use experimental evolution with both archaea and bacteria as a comparison to explore how adaptation and the emergence of trade-offs in these species. And so we've developed a system of halophilic archaea and bacteria that can grow together in the same culturing conditions. And I say they're halophilic to give you an idea of how much they really like salty conditions. Oh, the slides are slightly different. So if you were to grow 1 liter of, or if we were to make 1 liter of media to grow E. coli, for example, you might put in 10 grams of sodium chloride. But if you were to make 1 liter of media for these organisms, you'd put in 250 grams. So it's like near saturation point, very, very salty. And what this means is that when you put them on a plate, the plate will eventually precipitate salt, and they still remain very happy in these conditions. You can see some of the colonies even grow on the salt crystals themselves. So we have these 2 halophilic species. To explore the emergence of trade-offs, we grew them in 2 different environments, in rich media and in poor media. And the main components of these medias can be broken down into salinity, or the amount of sodium chloride, and the amount of resources in the form of yeast extract and tryptone, and then a concentration of other trace elements or other salts. And to look closer at the difference between these 2 medias, you can see that the rich media has higher concentrations of sodium chloride, as well as both of the resources, higher relative abundances, but has a lower relative abundance of these other salts. So what we did is we inoculated our replicate populations of archaea and bacteria in rich, and then propagated them in rich and poor media for 10 transfers, and then generated growth curves for the derived and ancestral populations. And once again, if this were like E. coli, or if it were the yeast species that I just talked about, 10 transfers would mean a 10-day long experiment. But even in the rich media, they're transferred every 5 days. And in the poor media, eventually was every 10 days. And so it was much longer, but only 10 transfers, and therefore only about 60 generations. So a very short period of time. Well, small number of generations. And what you can see is that, unsurprisingly, the ancestors grow faster in the nutrient-rich environment compared to the poor. It's an order of magnitude greater, so that's expected. Then following propagation in these 2 environments, we measured the derived populations in the selected environments, and you can see that after only 10 transfers, rapid evolution was observed in the poor media. So the bacteria that was selected in rich media performs about as well as the ancestors in the rich media, but the bacteria selected in poor media reaches higher densities compared to the ancestors in the poor media. We then switched the 2 medias, and so you can see the effects of selection become even clearer when you grow them in the non-selected environment. And you can see that selection in one environment leads to the emergence of very clear fitness trade-offs in the non-selected environment. So bacteria that was selected in poor media grows slower in the rich media, and vice versa. Bacteria selected in the rich media grows slower in the poor media. And this, so much so that one of the populations for bacteria in the poor media even lost the ability to grow in the poor media. And so this is like a very clear emergence of a fitness trade-off for the bacterial populations that happened very, very rapidly. Now I will show you the results for archaea. And if any of you have worked with archaea, you maybe would not be surprised to know that the story was not as simple for archaea compared to bacteria. And so you can see that, of course, the archaea ancestors reach higher densities in the rich media compared to the poor. But now both of the selected populations grow faster in the rich media and worse in the poor media. And so the effects of past selection differ significantly between archaea and bacteria, and the emergence of trade-offs was not uniform across species. So to try to understand the nature of these pleiotropic responses to selection that diverged across taxa, we then dig deeper into the different components of these 2 media to try to understand what was driving these differences. And the first thing is that when you look between the 2 medias, you have much higher growth in the rich media compared to the poor. And through pilot testing, we found that this growth disparity is mainly due to the differences in resources of these 2 medias. So we then took the poor media, added the equivalent amount of yeast extract and tryptone to that media, and found that growth was retrieved for the archaeal populations that are similar to what you would see in the rich media. To then distinguish between the effects of tryptone and yeast extract, we removed the tryptone from the media and found a moderate decrease in growth. So it had a moderate effect on growth. But the biggest growth difference was observed when we removed the yeast extract but kept the tryptone. So tryptone alone was insufficient to retrieve the growth that you see in the rich media. And the big growth disparity that we see is largely accounted to differences in yeast extract. We then took the area under the curve as a proxy for fitness. And you can see that growth and fitness depend on resource abundance. To a large extent, the abundance of yeast extract in these medias. So knowing this, we then looked at how knowing that there's a big effect of resource availability, we looked at the effect of selection in low resource and high resource environments and how that affects the niche breadth of these species across resources. So we created a resource gradient of yeast extract from 1 to 10 grams, measured the absolute fitness of the ancestors across this gradient, and then did the same for the selected population selected in rich media. And you can see that the selection response that we observed in just the selected environments is now manifested across this range of resources. So we not only see a response in one particular environment, but it also has shaped the niche of the species more generally. And in both cases, both bacteria and archaea that were selected in rich media have higher fitnesses across this range compared to the ancestors. We did the same for the population selected in poor media, and you can see that the same trade-offs that were observed for bacteria and the pleiotropic response to selection for archaea are now manifested also across this range and diverge once again. So bacteria that were selected in poor media have lower fitnesses across the range compared to the ancestors. And then the opposite is true, that archaea selected in poor media perform better across this range. For the archaea selected in poor media perform better across this range compared to the ancestors. So we clearly see the effect of resources on the adaptation of these populations and in shaping their niche breadth. We then wanted to look at the other environmental components of how they're shaping kind of the different edges of the niche, particularly the lower edge in low resources. How do the other components shape the response to selection? So how did selection shape niche breadth along the other environmental axes? So we grew all of the populations again in low resource environments, just 1 gram of yeast extract, but this time with varying levels of salt, either with or without tryptone. And the biggest effect that you can see is that for both bacteria and archaea, there's an additive effect of having high salt with tryptone. And so for all of the populations, they have there's a synergistic effect of both of these resources such that they are much higher in absolute fitness compared to all of the other conditions. So we see parallel synergistic effects occurring in the low resource environments. So you can, while these two species diverged in their response when looking at the resource gradient and the selected and non-selected environments, sort of the intricacies of these different environmental components at the lower edge remains parallel. But then the last component is to look at salt more generally. So we think that salt would be high salinity would be a strong selection pressure for these species. And so we wanted to look at how this shaped adaptation, both in low and high resource environments. And asked, how did salinity shape adaptation of these populations? So now we're at low salt and low resource environment. And now we're just looking at the concentration of these other salts, so whether or not it's present. So either you have poor media that has the other salts or poor media that doesn't. And first of all, for archaea, the presence of these other salts does not significantly impact the fitness of the selected populations. However, for bacteria, you see a clear genotype by environment interaction emerging. So now the presence of these other salts greatly impacts fitness in ways that vary based off of selection history. So the populations that were selected in rich media have a higher fitness when these other salts are not present. And then vice versa for the population selected in poor media, they have a higher fitness when the other salts are present. And this tracks to the selected environments that they had. So rich media, which does not have the other salts, the populations that were selected in rich media have a higher fitness when these other salts are not present. And poor media, which has the higher salts, populations selected in that environment have a higher fitness when they are present. So for bacteria, salinity appears to shape the nature of the fitness trade-offs in poor environments. But for archaea, salinity doesn't appear to be the aspect of the environment where selection was operating. This, of course, is in a poor environment. We then looked at the effects of salinity in a rich environment, high resources. So now we just have changes in sodium chloride. And once again, you see that for archaea, selection history does not shape plasticity of archaea. So regardless of whether they were selected in rich and poor environments, that both populations have a higher fitness when there's higher salt present. But for bacteria, another interaction emerges where the populations selected in poor media have a higher fitness when there's less salt present. And this once again tracks the amount of salt that's in the poor media. Populations selected in poor media have a higher fitness when that same concentration is present. So overall, you see that divergent plastic responses emerge, divergent across taxa, in response to salinity. For bacteria, in both cases, salinity appears to shape the nature of the fitness trade-offs in the poor and rich environments. And for archaea, this doesn't appear to be the aspect of the environment where selection is operating. So to summarize, fitness trade-offs and varying responses to selection emerge rapidly after only 10 transfers, or about 60 generations, for these species. The nature of these trade-offs are not only manifested in the selected and in one other environment, they're manifested across a range of environmental axes. And the same selective pressures can have very divergent outcomes across taxa, which can cause shifts in niche breadth that have important adaptive consequences. So looking back at extreme environments, you can see that by using experimental evolution with these halophilic species, we were able to understand how the confluence of these environmental components shaped adaptation in this particular extreme environment, which can occur very rapidly and lead to multidimensional shifts in niche breadth. Okay, so now that we have a system, we understand their patterns a bit more of adaptation. Let's go back to thinking about now if we evolve archaea and bacteria together, how could initial association have formed? So like I said, a big limiting factor of experimental work for archaea is the slow growth rate. However, in the Travisano lab, we have a unique position where Naui, the postdoc in the lab, propagated this species of archaea throughout her PhD and changed the time between transfers from every 45 days to every 11 days. They became evolved to laboratory or selected in laboratory conditions and began to grow faster. When she then came to Minnesota, we continued to propagate them. And so when we began the experiment, the time between transfers is around 5 days, which makes it a little more realistic to do experimental evolution and is compatible with the bacteria. We then established our long-term experimental aligns of bacteria and archaea separately in monocultures as well as together, using both settling selection and non-settling selection, with the possibility that settling selection could promote clustering phenotypes and potentially sort of physical association, interspecific association. We have continued the transfers for actually over a year now, over 100 transfers. So the data I showed you was after just 10. We've completed many more. And during that time, we've seen that the time between transfers has decreased even further from 5 to 4 and now every 3 days. And we've also observed several things during this time. So the first is that we periodically verified that in the co-cultures, the two species continue to coexist, which also was not obvious that a possible outcome would be that one competitively excludes the other. So they're still coexisting after many, many transfers. We also very early on began to observe differences in growth between our experimental and our control conditions, which seemed to indicate possibly a response to settling selection. And then we also visually started to see that some of the cells could settle in response to sitting on the bench. And this, if you take a culture of yeast, eventually all of the culture will settle to the bottom. But for these species, in the ancestral conditions, you don't see any settling after very long periods of time. So what also indicating perhaps that there's a response to settling selection. But both of these observations, both differences between the experimental and control and visual settling, have both emerged and disappeared throughout this long experiment. And so our current work is to go through our saved stocks, explore what's happening at those various time points, and to try to disentangle whether there's an interspecific association in our co-cultures, whether that's physical or metabolic. So that is the work that we have for looking at archaea and bacteria. I've also talked about origin of multicellularity. So now I'm going to take what we learned about these other systems and apply them to the origin of life. And I don't have time to introduce another experimental system. So instead, I'm going to describe how we're taking our previous knowledge of eco-evolutionary processes and applying them to the origins of life, and what we consider to be an eco-evolutionary approach to the origins of life. And this idea that eco-evolutionary processes are relevant during life's emergence is unfamiliar to a lot of people, but potentially not to you, because of the work of David Baum here at UW and colleagues in really doing a lot of work to demonstrate how ecology and evolutionary biology can help us understand how life emerged. So, as you're probably familiar, the origin of life is among the great open questions in science. And it is really a question of how you get from a non-living world to cellular life as we know it. And a key feature of cells, even the last universal common ancestor, is that the structures and the processes that make up cells are clearly derived. So they're the product of prior evolution. And so that means that somewhere during the emergence of life, adaptive evolution must have helped bridge this gap in complexity. And this might seem like a paradox, given that our understanding of evolution requires cells and genetic transmission. But it turns out that there are a lot of proposed theoretical and empirical systems that are evolvable prior to cells. Among these include RNA molecules or autocatalytic sets or protocells. And while these systems might seem very different from life as we know it today, we argue that the same eco-evolutionary processes are at play. So if you imagine a prebiotic adaptive landscape where the differences in peaks indicate different fitnesses, then you can imagine that any number of these evolvable systems could explore this adaptive landscape. And the particular chemical and environmental context that you're looking at in regards to their evolution would differ depending on which system you're working with. But a unifying requisite amongst all hypotheses of the origin of life, even those that seem to be at odds with each other, is that during the origin of life, innovation must have evolved. So if you have a simple beginning and a complex end, at some point between there, novelty must have emerged and greater complexity must have evolved. And so this question of how does innovation evolve is one that we're very familiar with. And that has been explored to some extent in extant life. And there was a recognition very early on that the similarity amongst these different transitions and complexity indicates that progress in understanding any one of them would help us to illuminate the others. And so what have we learned by studying these other transitions and complexity? Well, a key feature of these transitions is that the emergence of this new level of individuality deforms and transforms the adaptive landscape, which paves the way for further innovation. So the transition itself is an eco-evolutionary feedback. And this is what we demonstrated in the multicellularity work, that an eco-evolutionary feedback emerges as a result of having a new level of individuality. So you can conceptualize the origin of life as an eco-evolutionary feedback that is not just exploring this landscape, but is deforming the landscape, changing its possibilities for further innovation. And if you combine this idea, this cyclical idea of ecology and evolution, with the knowledge that stochasticity governs a lot of evolutionary processes, rendering it often unpredictable, then we begin to shift our idea of how to approach the origins of life away from viewing it as piecing together a stepwise progression of lifelike systems and piecing together the precise path that life took as it did on early Earth, because this is not only improbable given the limits and being able to predict past evolution, but it is also an unrealistic depiction of evolution as progress. And instead explore the several ways that evolvable systems might emerge and gain complexity, knowing that these eco-evolutionary dynamics are present throughout this process. So there must have been these eco-evolutionary feedbacks that change the adaptive landscape. There were likely extinction events or reversions to ancestral states, and key moments of not only competition, but cooperation amongst individuals. And so I have at least shifted my thinking about the origin of life away from trying to answer how precisely did life evolve on early Earth, and instead asked how can life emerge and gain complexity. So I'd like to thank members of the Travisano Lab for their help. Also in particular NAWI, the NASA Future Investigators Grant for funding the work, also the Early Career Committee for organizing this visit, and I'm happy to take any questions. Yeah, go ahead.
Hi, thanks a lot for the talk. I really enjoyed that you looked at salinity adaptation. That was really cool. I have two questions for you. One was about your definition of multicellularity. It seems like you're selecting for clumps of cells.
Yeah.
So why would you define that as multicellularity? Isn't it
Yeah.
just a clump of cells in biofilm?
Yeah, so it's not a biofilm, and it's not the coming together of separate cells to form clumps. That is a possibility that they could have done,
Right.
The flocking of individual cells. But these multicellular individuals have their own life history, and they only reproduce from then on forward as multicellular. So if you change the selective conditions, such as you're selecting for a shorter settling rate or a longer settling rate, what is selected on is the life history of the multicellular individual.
So I'm just curious about the design. So how do you evaluate that it's multicellular?
I guess for us, it's obligately multicellular in that it can no longer reproduce just as single cells, and it has a development period, like a juvenile period of smaller branches that form bigger, and once it gets big enough, it'll separate and form a new individual. If your definition of multicellularity requires cellular differentiation, you see that to the extent that there's apoptosis in some of the cells that will that it results from the benefit of the individual, but obviously not to that particular cell. But we don't see differentiation to a large extent of like different functions in different branches of the snowflake, for example.
Okay. Can I ask another question?
Yeah. Sure.
Okay, so in terms of, I thought the genotype by environment interaction that you found in bacteria, but not in archaea, it's really interesting. Sometimes we find that even between closely related species in our experiment. So would you say that that's a bacteria versus archaea contrast? I mean, different bacteria could also show or not show G by E.
Yeah, and they're different species in addition to different,
Right.
completely branches. I think that, so phenotypically, they're very, very similar. For a while, these two species of bacteria and archaea were confused for one another. They have the same rod shape and color, same salt in strategy found in the same environments, but phylogenetically, they're unrelated. So I think the explanation is the difference in that evolutionary history, whether it's because they're deeply diverged or not, we don't know currently. Does that answer your question?
Well, I'm just wondering, because sometimes when we do experiments, we'll find G by E in one co-I and not in another.
Yeah.
So it wouldn't necessarily be like the bacteria are in a context.
Right.
So I was just wondering to what extent you think it's because of that contrast, what kind of.
I think it's the difference in their evolutionary histories, but I don't know if it's because they're archaea versus bacteria or they're different species. Yeah.
Yeah.
Our follow-up work is to look at the transcriptomes of the species as well as genetic changes to kind of understand the actual mechanism of what's happening. Yeah.
So the question about the fusion of the K-lactis colonies.
Yeah.
When you have such a fusion, are they always among basically genetically identical clonal lineages, or do you observe fusions among unrelated lineages? In other words, do you get cooperation among clusters that are not clones?
Yeah, that's a good question. And you see cooperation amongst unrelated clusters. So within the cluster, all the cells are related. They're clones of one another because they're formed through not separating during budding. But different related multicellular clusters will still stick to each other. And in addition, you can see the emergence of unicellular free riders where those single cells will attach to the cluster and therefore have a free ride to the bottom of the cell and persist.
Also, when those unicellular individuals are also unrelated, right?
Yeah, right. It's a different genotype, I guess, depending on your
So in
definition.
other words, you don't have kin selection for these.
Yeah, I think that paper was about K-lactis and the flocking was with Will Driscoll as well as Mike Travisano, and that was a big finding, is that it's unrelated individuals that can have the synergistic association and therefore continue to coexist. And there's the idea of the free riders then is sort of a whole other experiment of like, is there cheating in the system where you're not contributing to the multicellular phenotype, but you still are gaining the benefit of being able to settle faster? Yeah.
There's lots I could ask about, wonderful. But one thing that's really interesting is this phenomenon of the presence of SRVCI slowing down the rate of evolution and multicellularity. And of course, that's kind of an odd phenomenon. You don't see that often.
Yeah.
I can't think of another example. So what are the hypotheses? And I can think of, I can think of 4, but I don't know which you think it is. Maybe the mutation rate, is the population size, is it selection differential, or is it generation time? Any idea which it might be?
My first thought would be population size, that having S. cerevisiae there is then decreasing the population size of K. lactis. You see the frequency is at a lower rate of unicellular, and therefore the frequency that the mutation happens is lower, and therefore it doesn't appear as fast and therefore is delayed. I don't think, because of the spent media experiment and showing that there actually is kind of potentially a cooperative dynamic between the species, I don't think that it's like excreting any, there's like a byproduct that's harming the growth of K. lactis. Yeah, that would be my guess.
I'm just wondering, as a follow-up to what David said, do the single cells take up nutrients from the other species more easily when they're single cells versus in your clone? Perhaps they can benefit more from their single cells. It's a greater surface area.
Yeah.
Contact with the bending.
That's a really good question, and I have not yet done the spent media experiment with multicellular individuals. I definitely could and will do that. But I would agree with your way of thinking that we already know there's a cost of being multicellular in general, because if you stop settling selection, they go back to unicellular. And that might be another cost, is that they can't benefit the uptake of the byproduct of the other as well. Yeah, that's interesting to follow up on. Yeah.
That was really cool. Very interesting stuff that I'm interested about, which is cool. I'm curious if you have a sense of, so your proxy, well, even not proxy, your way of looking at the evolution of multicellularity is cells that are dividing and not breaking. But you also suggest there are, kind of free writer cells that will attach, suggesting that there is a way that, unique cells can come together and clump.
Yeah.
Do you have a sense of what might have been more likely, or like the actual origin of multicellular?
Yeah.
If it was, something just not breaking apart, or things actually collecting?
Yeah. That's a really good question. And I think that previously it was more common to think that the coming together mechanism is more likely to form these clusters. But in that case, you have a higher likelihood of genetic conflict within the new individual. And therefore, that's sort of the whole framework, is it's hard to have, you have to have good cooperation within this new unit in order for there still to be a fitness benefit that allows it to persist. And so, at least for our system, and I think that it seems like it's a lot easier for it to happen, is if you have the staying together mechanism, because then you don't have the genetic conflict. Yeah.
Sorry. Do these guys have self, non-self recognition? Do you find that genetically related cells come together more readily than genetically unrelated cells? Is there any kind of recognition going on and more likelihood of clumping among genetically related individuals?
That is a good question. So it's only in K. lactis that you have the coming together clumping. In Cerevisiae, it's only the staying together mechanism that we see for multicellularity, as well as any sort of flocking, even though it does have the ability to flock in other settings. For K. lactis, I need to look back on that work a little bit more. I think that they have tried to explore whether it's more likely to happen amongst related and unrelated individuals. And I don't think that there is a correlation there. I think that it is just the stickiness of the cell, they're stickier, and therefore it doesn't matter if they're related or not. But I'm not sure. It's definitely an interesting avenue. Yeah.
So you're a Solibacter, I think, and a bacterium system. You mentioned that you're going to be doing transcriptomic analyses. But I was wondering if you have any thoughts as to whether you see any surface interactions, or is it just purely metabolic, some sort of metabolic complementarity? Because like if you're seeing precipitation, for example, like activating
Precipitation of the salt, you mean? Yeah.
Oh.
Sorry, what was that?
Yeah.
And you mean for the co-cultures association? If there is a difference between the settling co-cultures and the non-settling co-cultures, I would think the difference is a physical, like they've somehow stuck together. What we are exploring now that actually doesn't require transcriptomics is just separating the ones that have the archaea and bacteria that have been evolving in monocultures, and separating those that evolved in co-cultures, and then growing them together and separately to see if they've maybe lost the ability to grow separately, if they have a higher chance of being able to grow together, if they have that evolutionary history versus if they don't, and then plating them to see if there is higher growth when the two species are near one another. And that would kind of indicate that there's a metabolic association between the two. They've somehow become interdependent or potentially obligate if they can't grow separately now. That would be an indicator, and then we'd have to follow it up. In terms of which I think is more like, I think a metabolic association is more likely to occur without settling selection, just co-cultures together. But yeah, I'm not sure. If in the settling co-cultures, it's really unclear if right now, from what we know, if there is a physical association between cells that's causing them to cluster, if that is between the taxa, if it's just maybe they became multicellular, which is we've never seen that before, but maybe it has happened just within those species and they still aren't competitively excluding one another. We don't know right now. Go ahead.
Okay.
Your 5 minutes.
All right, so, when you looked at the selection lines, did you look at the genes under selection? Like, did you see a lot of ion transporter genes under selection?
Are you talking about for the archaea?
Yeah,
Or
they're bacteria under, high salt, low salt, different food conditions.
we haven't done any genetic work on it so far. It's just phenotypic assays. So we don't know why, if they have changed their strategy for combating the osmotic stress with the salty environments. We just see that there is a difference, and our follow-up work is to disentangle what the mechanism is for that. Yeah, that's our hope for those populations.
Curious to see what
We do have one question in the chat, which we'll read off from William Saucer. Any evidence of tight junctions or intercellular communication in the cluster organisms?
For the yeast work, I assume. I don't think so. I don't think that we have observed so far communication amongst the clusters. It seems to just be they're genetically similar or identical, and therefore remain in the clusters. But there's a lot of work that's been done on the snowflake yeast, and I'll look up specific if I'm wrong on that. But definitely interesting question. Okay.
I can ask, if you want a more philosophical question
Sure.
slightly. So you showed this diagram several times, like ecology, evolution, ecology.
Yeah.
Would you be open to the view that really there isn't a distinction, that evolutionary change is a kind of ecological change, that there's a broader category of change that we draw a sharp distinction between ecology and evolution, but that really that distinction itself shouldn't be viewed sharply?
Good question. I think that obviously the graphic I put up is a simplification of ecology and evolution. I think that in reality, oftentimes you can't disentangle between the two because it's so much of a feedback that the two are constantly there's constantly an interplay amongst them. And often if you're trying to observe an eco-evolutionary feedback discretely in a natural system, it's very difficult. In terms of like a broader, I mean, I know of a framework that understands it more generally. And I think that it's at that point, it's kind of a definitional difference. I tend to think that evolvable systems at the origin of life emerged very, very rapidly. And therefore ecology emerged just as fast because as soon as you have individuals that are interacting, you have ecology. And maybe that's like a caution to not kind of distinguish between the two when discussing the feedback, and kind of it's more the confluence of the two processes.
All right, if you don't have any more questions from the general audience, please join me in thanking Maria.